Hello, everyone. Welcome to this live broadcast, Overcoming Critical Barriers when Developing mRNA-based Cell Therapies. I'm Jeannie Linke Northrup, Managing Editor for Pharmaceutical Technology. We are pleased to bring you this expert roundtable sponsored by TriLink Biotechnologies. We have a few important announcements to make just before we begin with today's presentation. This webcast is designed to be interactive, and so we encourage you to ask questions throughout the event. You can submit your questions by typing them in the Q&A box, and that can be found at the bottom of your video player. You can also enlarge your slide window by clicking on that small icon located in the bottom right corner of the media player. Please note that all slides will advance automatically throughout the event. If you experience any technical problems viewing or hearing this presentation, please click on the question mark help widget, which is located in the top right of your presentation window. I would now like to introduce today's guest panel. Corey Smith joins us today as our guest moderator. Corey is the Senior Product Manager for Nucleic Acid Services at TriLink, where he currently oversees TriLink's mRNA CDMO service portfolio. Corey is dedicated to helping researchers and drug developers find rapid solutions to bring their mRNA drug candidates from discovery to commercialization. Also joining today's discussion is TriLink's Evan Myers and Khaled Yammout. Evan is the Associate Director of Process Development at TriLink, where he oversees the development of mRNA and plasmid DNA. Evan has extensive experience in this area, having held various positions in mRNA and oligonucleotide manufacturing, as well as GMP and process development. He's supported several drug substances. Evan's expertise spans biologics through synthesis, chromatography, scale-up, and process optimization, covering development, manufacturing, and analytical testing. And Khaled is the Senior Director of Analytical Services and Quality Control at TriLink, where he oversees all analytical aspects of method development and validation to product release and stability to support regulatory filings for both small and large molecules. Gentlemen, thank you so much for joining us today. Corey, please feel free to get started.
Thank you, Jan, for that introduction. Today, we're going to talk through some of the key differences between mRNA-based cell therapies and mRNA vaccines or other modalities. We're going to chat about some of the critical barriers that we have found and our customers have found when developing these mRNA-based cell therapies. Some of these differences include differences in scale, differences in the critical quality attributes and analytical profiling, as well as some of the downstream process differences, such as purification and other process differences. So with that, let's go ahead and get into some of the first questions on the panel. Question number one, what are some of the key challenges specific to mRNA-based cell therapies that you found when manufacturing? And we'll start with Evan. I'll pass this over to you.
Yeah, Corey, thanks for that question. I think when we think of mRNA-based cell therapies, we think of a need for, or a potential need for, really increased quality. Now, this could mean things like minimizing double-stranded RNA formation or reduction of double-stranded RNA. Could mean clearance of things like residual proteins, residual DNA. It could also mean just an increase in your, mRNA full-length product, or really just all increases in your overall mRNA quality. What this means from a process standpoint is really that when you focus on things like in vitro transcription, you want to minimize this double-stranded RNA formation as well as maximize your mRNA integrity while maintaining, a high-yielding process. But in doing so, you could affect things like your downstream purification. And so really focusing on that clearance aspect for things like a capture and affinity chromatography are now going to become much more important to clear anything that is needed in that synthesis reaction. So overall, the challenge comes with really needing to increase your quality and making sure that your processes can accommodate, but also provide the clearance needed for these, cell therapies.
Thanks, Evan. All right, Khaled, maybe from your perspective, from the analytical development and quality control areas, what are some of the challenges that you've seen throughout the mRNA cell therapy process?
Some of the challenges as this field grow and advance, there's an ask for tighter specs and also for analytical methods to be fit for purpose and more reliable to support the mRNA-based cell therapies. What that means is, traditional biochemistry and biological methods such as gel electrophoresis and all that, it's a good way to start, but eventually these have to move to more instrumentation to get an accurate reportable data to set specs and really challenge the detection limits. So as Evan alluded, you want to minimize double-stranded RNA. Well, how are you going to measure it? And how are you going to consistently figure out the low limit of detection that you it's not there anymore. So those are major challenges in terms of analytical techniques, and these technologies are evolving to characterize the mRNA. At the same time, the technology is growing for therapeutics and filing to be, as an IND or to get it to the market. So the race is on, and that is a major challenge to convert everything to truly not only quantitative, but also compliant in Part 11 and GMP compliant.
And just if I can build off that, are these some of the areas that you are expanding in your lab today? Are you working through some of this now?
Yes, we are. We actually brought our lab to full GMP, and we actually have GMP methods for sequencing using Sanger, and we're trying to figure out a way on how to capture the metadata and make it Part 11 for NGS, because for NGS to make it all the way to true filing and commercial, that hurdle have to be resolved.
Okay, so let's get on to the next question here. Looking back, obviously, I think both of you have had quite a bit of experience when it comes to manufacturing and characterizing mRNA. So there's certainly some lessons learned and insights that you've taken specifically when manufacturing for cell therapy, mRNA-based cell therapy. So let's dive into that. Khaled, I'd love to hear some of the lessons you've learned throughout this process from the analytical perspective.
Well, there's truly no true guidelines. Those guidelines are being evolved as we go. We've collaborated with USP, and we just, the second draft chapter for characterization of mRNA plasmid and LNPs was published about a month ago. And we're really working with a lot of agencies to actually solidify all these specific methods that's needed for that. But you also have to look at it from multiple perspectives. One is you're characterizing the DS, but also you have to characterize the drug product. And that's, for vaccines, you're talking about mRNA and the cargo with the mRNA, which is the LNP. Now, when you're talking about cell gene therapy, that's a different thing, because yes, you could have multiple mRNAs. You have the guiding strand, you have the mRNA, and those kind of like not only a DS, but also drug product if you're not taking it for further formulation and applying it straight to the cells. So that bring completely different challenge for the mRNA itself in terms of sterility, accuracy of the sequence, and also all of the DP methods in terms of cell-based to make sure it's working properly. So that's created a whole different challenge on that end. In terms of the DS, the challenge is again, to get the right sequence and get all the assays to be quantitative, and also have an understanding, how you apply all these to the different modalities. So the question always comes up, can I use a platform assays? Well, you got to define what a platform is. With that is most of the methods are UTR specific, they're sequence specific, and you have to figure out your tail and capping strategy. And there, the assays are very specific to that. So yeah, you can leverage generic methods to expand on them to your specific construct. You have to be really careful about that. So what we've been able to do here is we've developed generic methods. We applied them to over 50 constructs, and quite of these methods has been qualified and filed as an IND. So we are able to do all of that work for quite a few constructs.
And these constructs were completely different, had some similarities. I believe you mentioned some of these
Yeah.
are UTR specific methods, but I would imagine that each construct has its own unique characteristics that you have to apply these methods for. So these methods can be applied somewhat broadly, but still need to be construct specific.
Correct. So, to just to answer, to go a little bit deeper into that, the constructs vary quite a bit from length and composition, and even poly A tail length, different capping agents. So we're able to address all of that. Some of the things that you just have to, do more of a confirmation on is your methods that related to the process. Those things such as your residual NTPs, endotoxin, bioburden, concentrations, and the generic methods. Those you just have to do a confirmation to move forward with those from a generic method. But the critical one for characterization, you really need to pay attention to your sequence and how that could impact the assay.
Sure. Okay. Thanks, Khaled. All right, Evan, from the process and manufacturing side, I'd love to hear about some of the lessons and insights you've taken away from these projects.
Yeah. Thanks, Corey. And I completely agree with everything that Khaled is saying about, having a generic method or a generic process, but having to have the capability to do development on construct specific details. And I think that leads right to, one of my biggest lessons learned, which is really doing a lot of that upfront due diligence is super impactful and critical. And what that means from the process side of things is understanding, each step's critical process parameters and how those parameters interact with one another, but almost more importantly, how they contribute to these critical quality attributes. And I kind of mentioned it previously, but as these quality attributes change, or you want to focus on certain areas, understanding these process parameters and their impacts at each step, what's clearing here versus what's clearing there, what's forming and what's causing that to form, is really important. And what you can do with that piece of information is you can develop a really good platform process that you can, begin to apply to a wide range of constructs. We talked about different UTRs, different tail lengths, different, GC contents, different lengths of mRNA. If you understand these critical process parameters and these quality attributes and how they interact with one another, you can really begin to establish a good platform. Now, of course, there's always got to be room for construct-specific development or any platform technology to have slight changes, because it doesn't always mean that there's a one-size-fits-all for every single customer, every single construct, every single need. But with the understanding, you can take your platform and slightly change certain things, slightly change a buffer, or slightly change, your IVT condition in order to hit that critical quality attribute. So for me, it's understanding these processes and these parameters and how they're going to impact your quality as early as possible, I think is extremely valuable.
Yeah, so I'd like to pull on the thread of some of that development work that you've done. What have you seen is one of the more important areas to develop around? Would it be IVT reaction or purification conditions, kind of specifically for some of the areas where you mentioned before, highly pure mRNA suitable for cell and gene therapy or suitable for in vivo gene editing? What are some of those kind of critical process parameters you look at?
Yeah, and Corey, I think it's a great question, and I don't think that purification versus in vitro transcription or your enzymatic steps are necessarily more important than one another. I think they play to each other's strengths, or you have to have a purification scheme that plays to your IVT's weakness. But I do see that there's lots of potential in vitro transcription processes to generate, highly capped mRNA that is highly pure for full length product and low double-stranded RNA, all within your in vitro transcription reaction. And then your downstream purification processes would focus on, your removal of any residual protein or residual DNA or any just protein in general that might have needed to be put into those reactions in order to do that. So I think there's strategies, and there's really good strategies to solve a lot of these critical quality attribute needs in the transcription process, and then develop your downstream purification processes to pair with that to hit all your residual specs as well. So
well, it totally makes sense. I mean, kind of being able to leverage learnings from doing so many of these development runs, I think has definitely led the way for your team here. All right, I'll move on to the next question. When we're looking at, working with a partner or a CDMO for developing an mRNA-based cell therapy, what are some of the benefits that you guys see? And I think we touched on it a bit in terms of the expertise and how many projects you guys have worked on, but would love to hear some of the other benefits. And Khaled, I'll start with you from the analytics side. What do you think one of the main benefits of leveraging a partner in these situations would be?
Well, it will be everything under one umbrella, especially if you have the in-house capability for your analytical methods and you're not using a lot of contract labs. This allows you to adjust timelines, allow to support the customer to meet their timelines, critical timelines to conduct their clinical, their filing, and all of that. Also, in-house, if you have it built up to address all phases of clinical, starting from discovery to phase 1, phase 2 to commercial. If it's all done under one umbrella, you really don't need to do a lot of method transfer, a lot of technology transfer. Everything is within, and speed is important in that field. So if we all look at this from an mRNA standpoint, within 1 week you can get the mRNA made. If you have all your starting material, the plasmid and all this, it is really important to match the speed with the analytical. And if it's not under the same roof, then you could run into problems and delays. And also, if you actually establish the whole thing under one umbrella in terms of your starting material, your plasmid, your capping aids, and all the methods are developed in-house and tested in-house, if anything goes wrong, your investigation becomes really quick because you have all the data. You're not going back and forth between vendors and all of this. So this is really a key and very important thing regarding working with a partner that have a lot of flexibility and a lot of range of things to offer, from the starting material all the way to get you out the door.
Yeah. So you said something really interesting there that I liked about being able to leverage some of the same analytical methods and characterization methods from discovery through, of course, commercial. Does your lab, you guys have methods that would be able to fit for use for a discovery product as well as a phase 1 product, phase 2, 3, and beyond that you guys work on?
Absolutely. And we've proven that through a multiple construct, and we supported quite a few companies with their IND filing by giving providing the analytical method and qualification packages to help them with that. So, for us, we work with it as a collaboration. Those are customer specific methods for their construct, and they have a lot of input in terms of the write-up and the qualification. So at the end of the day, you're not only getting data, you're getting a long-term package to support your development program of a drug, which is extremely important.
Yeah, and I think the ease of transfer that you mentioned is extremely important, right? Being able to have confidence that your methods are applicable early up the funnel is very important, to know that you're characterizing your mRNA appropriately early on.
Yes, and we also shown that we're able to do it for so many different constructs. So we actually have the confidence that we can accommodate quite a few critical or challenging mRNA sequences to get what you need.
Sure. Okay, great. All right, Evan, from the process side, what do you see as kind of one of the best benefits of working with a partner?
Yeah, and I'd like to start and just say that I think, developing this partnership with a CDMO or a CMO in this, life cycle of a mRNA-based, cell therapy is extremely critical. It's going to help you overcome some of the challenges and coordination and communication pathways that you might have when you have many outsourced vendors. And really, it's going to help, with things like timeline, cost, and quality to have one vendor that you're going to rather than many. But when you're looking for a partner, you're looking somebody that has, as Cal's mentioned, in-house analytical capabilities that support, your mRNA, manufacturing. You're also looking for them to control your raw material sourcing, really how all the offerings that they provide that they can manage and take away from you. That's going to help on the quality side of things in the sense of auditing, one-stop shop. It's going to help with transparency and communication, so you're not going back and forth to get the answer. Cal had mentioned that if something happens, it's going to be a quick resolution. You're going to have all the information right away. But really, it's the support, and it's the process, and it's the entanglement of start to finish production inputs and outputs as well. And I should mention that at TriLink, we can support the mRNA-based cell therapy market through raw materials offerings like CleanCap, NTPs, enzymes, high quality plasmid, mRNA, and of course analytics to accommodate each one of those different raw materials as well as your mRNA. So really having that all in-house with the internal expertise is really going to be critical when you're looking for those partnerships.
Yeah, I think the simplified view of vendor sourcing is certainly one area that it helps to maintain a partnership. And then to Khaled and your point, having the expertise to handle not only of course the manufacturing of the mRNA, but the characterization of the mRNA, understanding that all raw materials are coming from the same spot is definitely a more simplified view of this process. So I think that sounds really good. I know we have one more question here, and I want to give you guys both a chance to answer this one. But really looking ahead to the future, what excites you guys the most, specifically about this market? Where is it going to go? How applicable is this going to be? I would love to hear your guys' response. Evan, I'll go ahead and start with you this time for this one.
Sure. I'm super excited about this market. I really believe that what we've experienced over the last few years is something that we're going to read, in history books, something that our kids are going to learn in school, maybe something called a biotech revolution. Just where it has gone and where it's going. I'm excited to see, because I really feel like the possibilities are endless. I'm really especially excited to see some of the potential for mRNA in the oncology space. I think it can make a huge difference there, along with many other end applications. But I think we've all been a part of it. It's going to be exciting to see where it goes next.
Totally, the breadth of applicability I think is incredibly promising. And Khaled, looking to you, would love to hear about, some of your experiences in the fields, stemming back as far as you've worked. I would love to hear about that.
So it is really an exciting field, and the benefits for the patients is just astronomical. Imagine in the field of oncology, you're going away from chemotherapy and radiation to something that is not very invasive, and you get the treatment. So this is really, this is phenomenal. The field has grown tremendously in the last 5 years. As a point, if you want to look at it from statistics, this is from the FDA website. There were approximately 50 related filings per year from 95 to 2015. That number has increased to 300 filings between 2016 to 2020. So this is indicating industry-wide trend toward development and implementation of cell gene therapy. And this shows the importance of this therapy and how it's bringing a huge benefit to improving patient life. And for me, this is what excites me. This is one step forward to really enhance the patient journey into recovery and normalcy in terms of life.
Yeah, a safer, more effective product brought to market. I think that's, from my perspective, again, the breadth of applicability, the safety of the method. I think that is really what excites me as well.
Gentlemen, thank you so much for an informative presentation. Before we begin with today's question and answer session, I would like to remind our audience how they can submit questions and be a part of the discussion. You can submit your questions by typing them in that Q&A box, again, found directly below the video player. Corey, go ahead.
Would like to get to a few questions that have come in from the audience. The first one, I know Khaled touched on a little bit earlier in the talk, but Evan, I'll give you a chance to answer first and then flip back to Khaled. Have you ever supported the manufacturing of a successful clinical batch of an mRNA cell therapy?
Yeah. The short answer is yes, we have. Khaled definitely touched on it, and I'll let him take kind of the wider breadth of this answer for sure. But yes, we've supported several successful batches for mRNA-based cell therapies, as well as their IND filings. I know Khaled's been very involved from the analytical perspective, but yes, we have.
From my end, if you've done a filing before, whether it's an IND or a BLA, large part of that dossier is analytical. And we set up our analytical service to support that. So that's why we encourage the customers to, understand that having construct and customer-specific method is very important, because we can share those with you. You put them in your filing, and we've actually supported that quite a bit. And not only that, we are also supportive of any questions that comes back from the agencies asking for additional data or clarification to help out the customer moving their program forward. So we've done this. I've done it in my past. I have over 35 years of experience, and I filed quite a few INDs and BLAs. So I fully understand that I'm trying to bring that to this modality. And
if I can just add on to that, to Khaled's point, and we had the question a little bit earlier about, when you're looking for a development partner, does having all the stuff in-house, under one roof, help? And I think this would be a great example of not having to go to all those different entities, to help with the support of the filing effort as well.
Definitely. Awesome. Okay. One more question came in around manufacturing processes and scales. What are the typical scales that you support or that you see the need to support specifically for this modality? Evan, I'll turn this over to you from the process side.
Yeah, of course. So I think, the scales can definitely vary dependent on that specific need or the end user, or really like the customer's testing. But, we've supported hundreds of mgs to tens of grams at the GMP level. I think it's very important to have a wide range of scales, to have, a discovery offering that's smaller scale and scalable into these hundreds of mgs and tens of grams. But, trialing covers a wide breadth of scale capabilities there.
Okay. That's great. Thanks, Evan. So another one came in around the analytical work. Khaled, what are some of the key challenges in determining mRNA purity?
Actually, that's a very good question. Before I answer that, I want to give a little insight regarding biologics. So about 40 years ago, antibodies became therapeutic antibodies for treating a lot of different diseases and all of that. At that time, if you ran a gel and you saw one band, that is your purity of that, and that was acceptable. Everybody laughs at that right now, but that's a really good example. Right now, if you're actually making an antibody for therapeutics, there's about anywhere between 4 to 6 impurity methods that you have to do, because each one tells you something different and different characteristics. Personally, I see the mRNA going into that direction. And we've already demonstrated that in our lab, that just one method, just whether it is a gel or a CE or HPLC, each one tells you something different. So at some points, you're going to look at all these orthogonal, determine the purity of your mRNA and the characteristic of your mRNA. I think the field will be heading that way in the near future, because the more we push the envelope on this modality, there's going to be some questions regarding how do you know your mRNA is as pure as you say it is.
Awesome. Okay, a couple more questions. I know we only have time for a few more here. Evan, I'll turn this over to you just for one more question, and then I'll finish it off with one last one. But Evan, question here about integrating raw materials into your production. Is this something that you guys offer? And if we have raw materials that we would like you to use, can you tech transfer those raw materials in?
Yeah, of course. We've tested many different raw materials, whether that's, wild type or modified raw materials, enzymes to NTPs to cap analogs. But if there's specific, raw materials that need to be tech transferred in, we've also accommodated that in the past. We can definitely put that within our processes. And we've done that and can do that routinely. So yes, of course.
Awesome. Okay. And last question for both of you. When you're designing these clinical mRNA-based cell therapies, what is the most important factor that you look to? And Khaled, I'll go ahead and start with you, and then I'll turn it over to Evan to bring us home.
So a couple of things one is the purity. The second one is the accuracy of your sequence. And what I mean by that is not just to say, I got, let's say, 95% coverage of the sequence using, let's say, Sanger sequencing or straightforward NGS. For cell gene therapy, I think you really need to figure out exactly where the bases are in the sequence, because those are really critical into repairing the damaged cells. And that is going to be, that's where this is also heading, to generate, to develop very accurate sequencing methods.
Awesome. Evan, to you.
Yeah, and Khaled mentioned that outside of sequence, it's quality, and we've talked about it before, but we're really seeing a trend within this market, around mRNA-based, cell therapies of what we can do to reduce double-stranded RNA to get, low double-stranded RNA in balance, as well as how can we get the highest levels of mRNA integrity in the form of, highest percent of full-length mRNA product. So really that quality, looking at your processes, looking at your analytics to test that, and then making sure that your sequence is 100% confirmed. So
awesome. Sounds great. I think we got a lot of good information. I know there's probably going to be a few more questions that trickle in, but we are out of time for now. Really want to thank the panel. Thank you, Khaled and Evan. And thank you to our hosts. I will turn it back to you, Gian.
As we wrap up today's discussion, I'd like to thank the audience for attending and for participating in today's event. Corey, a special thank you to you for moderating today's presentation. I'd also like to thank our participants and our sponsor, TriLink Biotechnologies, for making this educational webcast possible. Before we conclude, we'd like to ask everyone in the audience to participate in a brief survey. The survey will appear on your screen after today's presentation has concluded. You will also receive an email alerting you when this webcast is available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. Again, thank you to all for joining. We'll see you next time.